Hi. After this kind introduction, thank you for the opportunity to speak here, and I will speak about the comparing of atomic force microscopy with optical Fourier Ptychographic Microscopy, and this is for biological measurements. And I'm a postdoc in Professor Chris and Francis Lab in PDN Department, Cambridge University. And in the lab, we are performing a lot of mechanical measurements of our cells, and modern equipment must have the ability to give you a varying tissue stiffness measurement along with the imaging of the accompanying cellular changes. While you do have methods that are capable of doing it, and we saw a lot of them like the Bilal microscopy, traction force microscopy, pillars, etc., they are all limited. They need to know the exact materials inside. They are very sensitive to imaging noises, and this is the reason why an AFM, atomic force microscopy, that we saw earlier, is so appealing because it's a direct force measurement, and you can measure the adhesion, you can measure the topography, and it has a wide range of stiffness measurements. Modern FMs can produce measurements of 500 over 500 pixels in timescales of minutes, which is also appealing for biological processes, which are not so fast usually. However, there are factors of limitation. So first of all, the AFM head, but also for practical measurements, you'll probably use a gel or a substrate that you culture your cells on. And these gels that are usually of different mechanical stiffnesses will create aberration, and they will determine your working distance from the objective. This will really deteriorate your imaging. And also, many practical samples are grown on scattering substrates and even opaque surfaces, like stiff tissues. And the last thing is that usually in order to measure the right force, you'll need to glue a bead in the end of your cantilever, and this will create the very, very high resolution measurements that you thought you'll have negligible, so it will be with a lot less resolution. In order to lift those limited, we use the Fourier Ptychographic Microscopy, which is a method where you image, like in the presentation that was before me, from different angles, and this is done from the upper side of the AFM head, unlike the method that Pieter has shown in the previous presentations that he showed. You illuminate from different angles. Each angle in the Fourier plane creates a shift. So here you see a schematic of the system. You see a 1D Fourier schematics in the middle, and on the right you'll see a 2D Fourier plane. And this angle will give you a certain point in the Fourier plane, and the circle around it is the cutoff frequency that you can actually see with your imaging system. And then you will image from the center, and you'll have another point in the Fourier plane and another angle, and you just keep them overlapping enough so in the end you'll do a post-processing procedure that will give you enhancement of the resolution. It will also give you the phase, which actually implies of the index of refraction or the topography. So it might help the resolution of your, or just give you some feedback on the AFM imaging. It will eliminate scatterers, and even usually using this method, you can produce some topographic or tomography imaging. So you have a nice sectioning. I compared it to the ORX DSD, and as you saw that we also use the ORX DSD later on, so we can test our sectioning. And the nice added feature is that we get the pupil function of all the optical distortions. And then for fluorescence, for example, we could fix noisy images. So the first thing to understand is that when you use this method without the AFM head, you will probably have a different translation or even a bit of rotation in your Fourier plane. But when you put the AFM head on and you use the gels, etc., the whole sampling in the Fourier plane gets distorted, scaled, and even if you can see that you have like weird refractions from the AFM head. So first you have to calibrate that, and after a small process of calibration, we managed to attain beautiful images where you can see small features using the reconstructions that we're not seeing. And this is a 63x magnification with a numerical aperture of 1.3. Bear again in mind that the pixel size is 6.5. This will give you another limitations on your imaging. Moving to 20x with a lot poorer numerical aperture, you still see that our system manages to resolve beads. You can see on the left an AFM scan, high resolution AFM, no beads attached. In the middle, our reconstruction, and on the right, the white lamp that just didn't see the last bead. And again, bear in mind the pixel size, which is 6.5 micrometer. In this magnification, actually 2 pixels are just the numerical aperture. And we managed to attain that because the Fourier ptychography actually enlarged the plane. So you'll have, as a factor, here is a factor of 4 of the pixel. So the actual pixel size actually dropped by 4. And again, with the 20x, really poor images on the right from the white lamp, but our reconstruction really enhances the features. Also, like in the presentation before me, we could reconstruct the compartments and the vesicles inside the cell. So on the left, you can see a Hoch stain of a human glioblastoma cell. And in the middle, our reconstruction, and you can see how it follows the same compartments inside the cell. And on the right, the white lamp, which does not show them. That way, we managed to attain around 1.56 numerical aperture of a 1.3 numerical aperture objective. And as I mentioned before, we have the reconstructed pupil function seen in the middle. On the left, you can see this is a cortical neuron cortical cell of a rat, and its membrane is dyed with RFP. And we also incorporated the 200 nanometer beads. So the whole thing sits on a gel with beads, and you can see that the imaging of the fluorescence imaging is rather noisy. Deconvolution with the pupil function, and you'll have a very nice image, and you will reduce the noise significantly. If I had the chance, you could see nice 3D reconstruction images of the fluorescence. What about the quantitative measurements of the topography? So again, if I know the index of refractions of the materials, I can use the phase, and then I could reconstruct topography of the surface. So here we see 1 micron spheres, polystyrene spheres. And without the AFM, we could reconstruct their height. We did it for 1 micrometer, half a micrometer, and 200 nanometer beads. And you can see that you can reconstruct the topography of the bead quite nicely. However, when you introduce the AFM, things are really broadened, and the standard deviation also broadened. However, this could still give you a quality and somehow back your data, and it will give you some more information that you may want to use. And what about scattering mediums? So here I introduced a sample that really put a lot of spheres. They were just everywhere. And you could see this is, again, the human glioblastoma cell. And our reconstruction could show you fine structures inside the cell. that you just cannot see because of the scatterers that are just preventing you from really imaging them. And again, if I had the chance, this is an ORX DSD reconstruction with a pinhole of those scatterers. And here I also show a profile that could that you could see and assess the imaging quality. And on the right, it's the fluorescent beads. There are beads here that are 200 nanometers and 1 microns, a lot of jungle. It was a jungle in the scatter. And with that, I would like to thank my group and the Blavatnik Fellowship that supports me. Thank you for listening.
Thank you so much for a great talk. Yeah, I'm missing coming into the Franz lab. I've been talking to Alex online about various things recently, so hopefully I'll see you again soon when we're allowed to visit. So does anybody have any questions for Dr. Wagner? We could put it in the chat folder. We are running very short of time. I just had one quick question. You were saying about the noise in your images. Where is the noise coming from? Is it shot noise that's in your sample itself, or is it coming from something else?
Yeah, so it could be a stray light. So, for example, there the dyeing was not so good. A lot of time you have a small like surfaces of fluorophores just staining a compartment in the gel, so it's somehow sticking there. You'll have a lot of stray light in that case. For the method, it does not really matter. So because of the high sectioning, you are preventing or you actually averaging this background noise or stray noise. Mm-hmm. And also because you have from each location or from each angle, you have another shot or another prediction, let's say, you can also average the statistical noise, if that's what you meant. Yeah.
Sorry, I just froze for a second there. Yeah, thank you. And what method were you using for your deconvolution? Was it a proprietary algorithm, or is it something that you did yourself using the beads?
So everything here is things that I did by myself, but you can use once you'll have the point and function of your pupil, then you can use. There are so many methods there.
Yeah, absolutely. That's why I was kind of. Did you use one that has a specific name so people know what to use in their work? But obviously, yeah, there is a multitude.
A Wiener estimator, a normal Wiener estimator for the deconvolution. Of
Sure.
course, then you will have the problem of optimizing your parameters, etc., etc. But now I'm waiting for the research, and then I'll determine exactly the methods and the properties.
Sure.
Very specific.
Great. Well, thank you for sharing your work with us. I really appreciate it, and I hope to see you at the university when we're allowed to meet again.